All right, so welcome back, everyone. So what we're going to look at here is we're going to talk a little bit about immunological memory. We're going to talk about kind of come back to antibodies just a little bit, talk about different types of responses, do a little discussion on vaccines, and then we will be done. So this idea of immunologic or immunological memory is the idea that after we've encountered an antigen, right, and we undergo that process of clonal selection, they become activated. When they activate, right? So when our T cells activate and when our helper T cells and cytotoxic T cells, right? Some of them become the active form of the cell, so activated cytotoxic T cell and activated helper T cell, and then some become the memory cell, right? And remember, the memory cell, it retains the same receptor specificity as that original cell, right? So the naive cytotoxic T cell has some receptor specificity against some given antigen. And whenever it becomes activated and divides into an activated cytotoxic and a memory cytotoxic, both of those retain the exact same receptor specificity, right? So those memory cells are present in the event that you encounter the same antigen again in the future, right? Now, whenever you're talking about the idea of antibodies, right, the antibodies that are produced by B cells, or really by the plasma cells, right, those circulating levels of antibodies, those antibodies also have the same receptor specificity. So in the event that you have this initial response, this primary response, in which you are initially exposed to an antigen, you produce antibodies, and you can kind of go through these latent and lag phases where it's several days, no antibody detection, and then the antibodies begin to be produced, so IgM, IgG. Those antibodies that are produced have the same specificity as maybe the original antibody that bound a particular antigen on the surface of a B cell or something like that, right? Now, where the memory comes in is whenever you have your second and your third and your fourth and your 15th exposure to that same antigen, right? The the antibodies that are produced, not only are they going to be the same, right? They're going to have that same specificity, but they're going to also typically be secreted in higher levels, right? And also much faster as well, right? So that latent or lag phase is much shorter than in the primary response because you have those memory cells, right? You have those same cells that were produced during the initial encounter. Those are there whenever you have your second encounter and your third and your fourth and so on, right? And so those antibody levels are going to rise very, very quickly, okay? So again, with subsequent antigen exposure, the memory cells make more contact with the antigen, they produce more antibodies, the secondary response is typically greater, and vaccines are effective in developing that memory, right? And we'll talk about vaccines here in just a little bit, right? And so with our different types of responses, right, so we'll have a primary response, a secondary response. In the secondary, typically the pathogen may be eliminated before we even notice, right, because of that very quick and drastic and significant rise in antibody levels, we may not even notice it, right? And so this is relevant in kind of our very immediate context with the ongoing measles outbreak, right, in Texas. And so if you have measles, if you've had the measles vaccine, the MMR vaccine, you likely won't have it again, even if you're exposed, because you've had this initial exposure, and then you have the memory, right? You have those circulating antibodies present in the event that you are exposed again, right? So it's not that you can't be exposed again, but you're likely not going to have the disease again, right? And so this here is showing the differences between that primary and the secondary response, right? So a few things I want you to notice. One, I want you to notice the the lag time between the primary and the secondary response, right? So this blue shaded region, that's the lag time, or that latent phase, as it was called. Um, that is longer in your initial response, in the primary response, because you've never been exposed to that particular antigen before. So of course it's going to take longer for your immune system to see the antigen, recognize it, bind to it, activate it, activate the cells, and then initiate the response, right? So it may take up to 3 days, right? 3 or 4 days. In the secondary response, notice that the lag time is very short, right? Maybe a day, maybe not even, right? So that's one thing to pay attention to. The second one is the how quickly you get the, or not how quickly, but the significance in terms of the amount of antibodies that you have, right? So in the primary response, right, you release some IgM, some IgG from your plasma cells, B cells. In the secondary response, it is significantly higher, right? So again, that secondary response is much more drastic. It's much more exaggerated than in the primary response. Okay, so as we mentioned, vaccines are a helpful way, or a helpful way to kind of bring about this memory that we're talking about, right? So vaccines, they're going to contain either a weakened, a dead portion of the microorganism or the virus or whatever it happens to be. There are ones now, we'll talk about, like the COVID-19 vaccine, right, which is actually not, it doesn't contain the virus, it doesn't contain a weakened version of the virus. It is actually a what's called an mRNA vaccine, and that's a different type of technology than what you get in, like, say, the MMR vaccine or other ones that have maybe a weakened version of the pathogen or a particular protein of the pathogen, things like that, right? And so these are stimulating our immune system cells to develop these B lymphocytes. They then produce the antibodies. Those antibody titers are going to circulate. Now, in a more broad context, right? So kind of at a population level, you can talk about something called herd immunity, right? And so herd immunity is this idea that if you have a significant enough number of people who are vaccinated against a disease, then they will prevent the spread of the disease to those who are not vaccinated, right? So essentially, think of it as being the virus or the bacteria, whatever it is, the disease is unable to be transmitted from person to person to person, ultimately to those who are unvaccinated, because such a high number of people are resistant to the disease, right? They're vaccinated, right? And so typically you need a high number here, so maybe 85 to 95%. In the context of this kind of ongoing measles outbreak, most of the discussions have been around, herd immunity being somewhere around 95%, where in the affected areas in Texas, the vaccination rate was much lower than 95%, especially among certain kind of community groups, right? And so if you don't have that herd immunity, you don't have a high enough vaccination rate in the population, then the spread of the virus, in this case the measles virus, is going to be much more severe, right? And so vaccinating yourself not only protects you, but it's actually protecting other people as well, right? So vaccination is an extremely important and extremely necessary public health measure, but also individual health as well, right? So there are various ways that you can obtain immunity through vaccines and otherwise, and these are called active and passive, right? So you can read through this, but I'm going to show you this graphic here because I think it kind of breaks it down a little bit better, right? So active and passive, they differ in terms of whether or not you make your antibodies or you are given your antibodies, right? So active immunity, you make your antibody. You actively make them, produce memory cells due to contact with an antigen, right? Passive immunity, no production of memory cells, antibodies from another person or an animal, right? So meaning you are given the antibodies, you don't make the antibodies, right? So for both of these, you can have natural and artificial versions, right? So you could have antibody therapy where you're given antibodies. This would be artificial, passive artificial. You could have naturally acquired antibodies through the placenta and breast milk. So this would be passive natural. You could be given a vaccine in which you stimulate the production of antibodies, so that's artificial, but it's active. And then you can have natural active, and this is just a normal infection, right? You're out and about in the community, the environment. You get the flu. You develop your antibodies against it. That's natural active immunity. Right. So for these vaccines, again, artificially acquired active immunity, there are what are called inactive vaccines, in which you have a kind of a destroyed version of the pathogen that a destroyed version of the pathogen that's not going to give you the actual disease, but it's going to stimulate your immune system to produce the antibodies against whatever that is, right? So in this case, you may need further booster shots for to retain the immunity, right? So these are things like Hep A vaccine, flu, polio, rabies, things like that. You have what are called live attenuated vaccines, where you kind of have a weakened version of the pathogen. And these are typically more long-lasting, right? So the MMR vaccine, the measles, mumps, and rubella vaccine, is a live attenuated vaccine, right? So provides much more long-lasting immunity, right? You can have recombinant and conjugate vaccines, in which you kind of use a piece of the germ, so just a particular portion, so a protein, a sugar from it, but they still give you a very strong immune response. So shingles vaccine, HPV vaccine, hepatitis B, these are all examples of these recombinant and conjugate vaccines. And then kind of newer ones like the COVID-19 vaccine, this is an mRNA vaccine. So you're not getting the virus, you're not getting a part of the virus, you're not getting a protein from the virus, you're getting a strand of mRNA, right, from the virus, or that would be found in the virus. And so this mRNA, remember mRNA codes for proteins, right? And so the mRNA is then read by our own cells to produce the viral proteins, and then our immune system responds to those viral proteins, okay? And so you're not actually getting the virus. There's no chance of you getting COVID because of the COVID-19 vaccine, because you're not actually getting the virus. All you're getting is a little strand of DNA that would produce proteins found on the virus, and then our immune system recognizes those and combats an immune response against them, right? So various types of vaccines that you can look at, right? So kind of last few things that we'll look at here are some clinical views. So hypersensitivities, again, these are just exaggerated immune response in response to some type of antigen. So acute hypersensitivities, subacute within a couple of hours, and then delayed over several days, right? So you could think of various types of maybe allergic reactions, right? So allergies caused by some type of allergen. So common ones you think of are pollen, latex, and peanuts, but there's dairy allergies, right? There's allergies to various types of fruits, right? There's lots of different allergies, right? There's gluten allergies, things like that. And so in this case, you'll have a sensitization phase in which you're initially exposed. You'll then have an activation where the immune cells and the immune system, inflammation, things like that are activated, and then you'll have an effector phase as well, right? So you can think of all of these different symptoms that you would expect to see in an individual with an allergic reaction, right? Now, some allergic reactions are more severe than others, right? So you can have relatively mild allergic reactions in which you get runny nose, watery eyes. You know, so if you're allergic to dogs or cats, right, you may kind of get itchy. You may have some watery eyes, runny nose, but it's not, you know, it may not be all that dangerous. Whereas you can have severe allergic reactions, and you can end up in something called anaphylactic shock, in which you get major vasodilation, inflammation, blood pressure drops way down. Airways can constrict, right? So breathing becomes difficult, and these are cases in which you would need maybe something like an EpiPen, right? So to treat this with epinephrine, right? And so in those cases, those are extremely dangerous, potentially fatal hypersensitivities. Not just one that would occur, you know, if you accidentally rub up on a dog and you're allergic, something like that, right? These are very severe allergic reactions, right? And then kind of the last thing that we'll look at here is HIV and AIDS. So we alluded to this earlier, right? And so AIDS is acquired immunodeficiency syndrome, and HIV is human immunodeficiency virus. So what this is HIV is the virus, And then AIDS is the disease that can occur from infection with the virus, right? So you can have HIV without AIDS, but you can't have AIDS without HIV, right? And so this can be transmitted through bodily fluids, so through sexual intercourse, needle sharing, and also potentially breastfeeding as well, right? So combating each of those potential kind of transmission modes is helpful in reducing the transmission of HIV and thus AIDS as well. Um, so the HIV virus, right? HIV itself acts on and targets the helper T cells, right? So if you target and destroy the helper T cells, then you are now acting against both the cell-mediated immunity and the antibody-mediated immunity, right? So in other words, the activation of your cytotoxic T cells to destroy infected cells, abnormal cells is diminished or decreased, and then your ability to produce activated B cells and plasma cells and produce antibodies is also decreased as well, right? So big thing here is that you are severely immunocompromised, right? And so oftentimes individuals who have HIV and they also have AIDS, is their cause of death is usually associated with an infection called an opportunistic infection. And these opportunistic infections are those infections that typically you would not get if you didn't have the underlying disease, right? So maybe some type of fungal infection or some type of bacterial infection, right? So you are immunocompromised to the point that you get a disease that otherwise you wouldn't get, or otherwise it would not be all that dangerous, but now because you're immunocompromised, it can potentially lead to severe complications, including death. Okay, so there are therapies for HIV and AIDS. So there's something called HAART, HAART, highly active antiretroviral therapy, in which medications are given to help reduce the viral load. So in other words, the amount of the HIV virus that's found in the body. But these can be very expensive as well, right? So they're not always a treatment for individuals with HIV and AIDS, but they can lower the viral load to the point that it can no longer be detectable and transmitted during sexual intercourse, things like that, right? And so just a few things to think about, right? So we're going to stop here, and if you have any questions about adaptive or acquired immunity, please feel free to reach out.